With nothing further, I really would like to present to you Professor Mary-Ellen Harper. She's a professor at the University of Ottawa and the research chair in mitochondrial biogenesis. Dr. Harper's research focuses on mitochondrial energetic mechanisms that impact the efficiency of energy conservation pathways in mitochondria. Changes in the efficiency of energy conservation can affect oxidative stress and in cell signaling, thus affect the development of many diseases. Experimental approaches span from molecular in vitro studies to mouse models and to integrated studies in patient populations. She and her group has published enormous amounts of papers, 195 peer-reviewed papers, and has more than 17,000 citations. Her research is impressive and is currently funded by several mechanisms, by CHR, NSERC, the Heart Stroke Foundation of Canada, Canadian Nuclear Laboratories, and the National Research Council of Canada. Dr. Harper also holds a university research chair in mitochondrial bioenergetics. She is also, the director of Ottawa Institute of System Biology and the director of the NSERC-funded Metabolic Advanced Training and International Exchange Training Programs. It's her work and her research led her to be the 2021 awardee of the Canada Lifetime Achievement Award in Diabetes. That's one of the most impressive and prestigious awards. Congratulations, Dr. Harper, and thank you for joining us. The floor is yours.
Thank you very much, Anna. It's a real pleasure and an honor to be here. Let's just see here if I switch over to presentation mode here, if I can see the slides. I can't see the slides yet. Just in a tiny, tiny screen there. Let's see. Yeah. Slides. Sorry for the delay here. I just can only see the all of the contributors here. Okay, I think I've got it. Okay, awesome. So sorry for that delay. Okay, so, and I'll advance the slides on the cell phone here, I guess. It's a cool new system. So yeah, thanks again. Sorry for the delay. It's a real honor and a pleasure to contribute to the second annual symposium here, and it looks to be a very exciting program, and I'm looking forward to hearing many of the presentations. So as Anna mentioned, I'm going to be talking about some fundamental mechanisms that impact the efficiency of energy transduction in cells. And this has implications for energy expenditure of cells, for redox, for ROS, for cell signaling, and so has implications for many disease states. And most of the translational research that we do here in my laboratory at the University of Ottawa focuses on obesity and diabetes, with some other collaborations as well. And in the latter half of my talk today, I'll talk about that translational research in obesity and diabetes. So as you all know, this is an exciting time for mitochondrial biologists, as then there has been a real resurgence of interest in these dynamic organelles. Our research, again, focuses on bioenergetics, ATP synthesis by oxidative phosphorylation in mitochondria, And these reactions and pathways in mitochondria also have significant implications for many of these other functions in mitochondria as well. So, of course, mitochondria are the metabolic hubs in cells, and they receive the electrons or reducing equivalents from the stepwise oxidation of fuels in cells, and then channel that potential energy into the synthesis of ATP through oxidative phosphorylation, allowing an astounding amount of ATP produced by the body per day, upwards of 70 kilograms of ATP produced in the body per day. ATP is not stored within cells, and so mitochondria have to quickly adapt to the ATP demands of cells on a moment-by-moment basis. And these processes of oxidative phosphorylation in cells rely, of course, on oxygen. And that means that our bodies require oxygen, about 550 liters of pure oxygen per day. That's a lot of air intake to support ATP production in mitochondria and cells throughout the body. And this oxygen requirement for oxidative phosphorylation is the basis for the respirometry approaches that so many of us use in our laboratories, whether it be Clark-based electrode type technologies, including the high resolution respirometry approaches, the seahorse approaches that many of us also use, and also at the whole body level, indirect calorimetry measurement of metabolism in our mouse models and in our translational work in our human participants. And I am looking forward to the technical mitochondrial technology session later today that Chris Perry is giving for the trainees. I think that's going to be fantastic. Okay, so again, cellular ATP is not stored in cells, and it's tightly regulated over a quite narrow range, 2 to 5 millimolar in cells. And so mitochondria need to match ATP production acutely to meet ATP demands. And some of the major consumers of ATP are shown in this slide. And in the last point there, cell-specific type ATP demands, including insulin secretion by pancreatic beta cells, muscle contraction, neuronal firing, and the synthesis of urea in liver cells. So this slide shows some very familiar diagrams, I'm sure, from Leninger textbook of oxidative phosphorylation on the right-hand side. And the electron transport chain there receives the electrons from those stepwise oxidative reactions in the cytoplasm and also in the mitochondrial matrix. And those electrons or reducing equivalents provide sufficient power for complexes of the electron transport chain, complexes 1, 3, and 4, to pump protons out of the matrix into the intermembrane space, producing proton motive force. And that proton motive force is then used to drive the synthesis of ATP through ATP synthase. And you can see the return of protons through the FO subunit of ATP synthase in the mitochondrial inner membrane. You can also see the site of oxygen consumption at complex 4 of the electron transport chain. So the oxidative phosphorylation supplies about 90%, generally speaking, of a cell's ATP, with about 10% of the ATP coming from glycolysis. But of course, this depends on metabolic state and cell type to some extent. And these are some of the things that we measure using seahorse technologies and specific assays. Now, this slide shows a video clip from John Walker's group at the Mitochondrial Biology Unit at the University of Cambridge. And it really does show, indeed, that ATP synthase is literally a molecular motor receiving ADP, inorganic phosphate, and producing ATP when protons move through the FO subunit in the mitochondrial inner membrane. Now, most textbooks would still have us think that there is a perfect coupling between the oxidation of fuels, and in this table here, it's glucose, that the full oxidation of glucose leads to 30 or 32 molecules of ATP through oxidative phosphorylation, 30 or 32 depending on the shuttle mechanism to get the electrons into the mitochondria. But the efficiency of oxidative phosphorylation is far from perfect, and fuel oxidation is not always coupled to oxidative phosphorylation. And in fact, there's great variability in oxidative phosphorylation efficiency, but whether you refer to that as the amount of ATP per unit of oxygen consumed by your cells or mitochondria, the amount of ATP per unit of glucose or fatty acid oxidized, that we usually obtained far less ATP per unit of fuel oxidized. And the main reason for this is a leak of protons through the mitochondrial inner membrane through various complex mechanisms that I'll talk about in a few minutes. But the implications of protons leaking back into the matrix bypassing ATP synthase is that there's decreased mitochondrial membrane potential or proton motive force, meaning decreased or abrogated ATP production, and continued oxidation of fuels and oxygen consumption in the absence of ATP production. Now, this slide shows it from a different diagram. This is a modified diagram from Jose Enrique's lab in Spain, which I really like. And it shows that proton leak uncoupling is, researchers found that proton leak uncoupling is ubiquitous. It occurs in all cell types in which it's been studied, all cell types throughout the body, and it's energetically important. And it's been estimated that 25 to 50% of resting cellular energy expenditure is due to proton leak uncoupling. Now, this seems like a large amount, and it is, and it seems potentially, seemingly wasteful, but we'll talk about the physiological purposes of proton leak in just a few moments. Now, importantly, when ATP demand of a cell increases, for example, in a contracting muscle cell, the leak of protons decreases and the return of protons to the matrix return to ATP synthesis. So the efficiency of ATP production can change depending on ATP need. Now, the most pronounced and widely understood form of mitochondrial uncoupling is that in brown adipose tissue. And the physiological function of proton leak uncoupling in brown adipose tissue is thermogenesis for thermoregulation. And the protein responsible for this uncoupling, UCP or UCP1 or thermogenin, was identified by David Nichols back in 1978. And the idea that thermogenesis in brown adipose tissue could have an impact on risk for obesity was the idea was first put forward by Canadian or University of Ottawa researcher Jean Hims-Hagen, who published this hypothesis back in 1984 in the New England Journal of Medicine. And sadly, Jean Hims-Hagen passed away in this last year. It was in the late 1980s that I joined her lab as a PhD student. And then eventually in 1985, the gene for uncoupling protein 1 was identified. Now, the findings that uncoupling occurs in tissues outside of brown adipose tissue was first noted by Peter Mitchell in the 1960s. And Peter Mitchell was awarded the Nobel Prize for the discovery of the chemiosmotic theory of oxidative phosphorylation. And they noted this proton leak in their isolated mitochondrial preps, but it was generally interpreted as damage to the mitochondria as they were being isolated. But these findings were picked up and followed up by Martin Brand in his photos at the bottom left there, And in the late 1980s, he showed that uncoupled oxidative phosphorylation occurred in hepatocytes and thymocytes, and that this proton leak uncoupling in various cell types was not due to mitochondrial damage and was a regulated process within cells. And I was fortunate to join his lab as a postdoc in the early 1990s. Then there was an exciting period of uncoupling protein research in just after 1997, when the novel uncoupling proteins were discovered. And since that time, a lot of research has continued to try to elucidate the real functions of these proteins and other potential mechanisms controlling this mitochondrial proton leak. Now, this slide shows the 6 purported uncoupling proteins and their tissue localization. And then in the right-hand side of that table, the approximate homology of these novel uncoupling proteins to the original UCP1 in brown adipose tissue. Now, all of these uncoupling proteins belong to the mitochondrial carrier superfamily of 50. There are 53 proteins within this SLC25 anion carrier protein family. And it's been suggested that some of these uncoupling proteins, the novel uncoupling proteins 2 to 6, perhaps be renamed because these proteins, like many of the other SLC25 proteins, have pleiotropic transport characteristics and transport other molecules, perhaps beyond protons. And it's also important to remember that the expression levels, the protein levels of UCPs 2 to 6 are 2 to 6 orders of are about 2 orders of magnitude lower than that of UCP1 in brown adipose tissue, where UCP1 represents quite a remarkable 10% of mitochondrial protein. So the uncoupling impact of these other novel uncoupling proteins can be less lower as a result of this. So a lot of research has occurred since the original discovery of the novel uncoupling proteins, and a lot of research has delved into what the mechanisms might be, why such a apparently wasteful mechanism could occur in cells, what its physiological function might be, and what the disease implications are. And at the bottom there, I've just indicated that there have been over 8,000 published papers in this field since the identification of UCP1. Just a few more words about UCP1 in the context of metabolic health. There's no question that UCP1 and brown adipose tissue thermogenesis is very important for thermal regulation, but the health impact of brown adipose tissue, or the disease implications, have been controversial. And I've just included these results from Betcher and colleagues published last year in Nature Medicine, showing that those adults with detectable active brown adipose tissue, and the data are shown in the brown bars here, compared to those with little to no active brown adipose tissue, that those with active brown adipose tissue have a lower risk of type 2 diabetes, dyslipidemia, coronary artery disease, cardiovascular disease, congestive heart failure, and hypertension. And these protective effects are more pronounced for those with overweight and obesity. And you can see the protective effects on fasting blood glucose and triglycerides at the bottom there. Now, the physiological roles of the novel uncoupling proteins, and particularly UCP2 and UCP3, where there's been a lot more research than the other proteins, have been many and varied. And it's been suggested that the common underlying mechanism for these various physiological functions is through the control of redox and ROS in cells. Now, how can proton leak uncoupling impact the production of reactive oxygen species and redox in cells? Well, proton leak causes a decrease in mitochondrial proton motive force as protons return to the matrix. And so that decreases the back pressure of protons across the membrane, which facilitates proton pumping at complexes 1, 3, and 4. And coincident with proton pumping is electron flow. And if electrons continue to flow within the electron transport chain, they tend not to escape and interact with oxygen to produce the most proximal form of reactive oxygen species, superoxide, which can then go on to produce potentially damaging species of reactive oxygen species. Now, so it seems that some of these slides are a little bit mixed up, but hopefully you can understand. Reactive oxygen species also play important positive roles in cells. They play important signaling roles in cells. And the form of reactive oxygen species that is most important in the signaling within cells is hydrogen peroxide, because it has a longer half-life and because it can traverse membranes within cells. At low levels, hydrogen peroxide can function in this capacity, signaling and adaptation through the modification of protein thiol residues. However, with sustained and higher levels of hydrogen peroxide, this can lead to further irreversible modification of cysteine thiols from sulfenic acid residues to sulfinic and sulfonic residues, damaging the activity of those proteins. However, in the top right, you can see that the lower levels of hydrogen peroxide can act in signaling capacities through reversible post-translational modifications of those thiols through glutathionation in the top right there, reversible, for example, through the activity of glutaredoxin 2, or through disulfide bridges internally or between proteins, which can be reversible through the activity of thioredoxin 2. Now, the molecular mechanisms of proton leak remain poorly understood beyond levels of proteins within the mitochondrial inner membrane. It's been shown that the post-translational modification of UCP1 and UCP2 and 3 are important as well. And the work of Ed Ciucciani and colleagues at the University of Harvard showed that the sulfenylation of certain cysteine thiol residue in UCP1 can augment the uncoupling activity of UCP1 in brown adipose tissue. And research in our laboratory with some work from former postdoc Ryan Mayu and others, we found that those homologous residues in UCP2 and UCP3 could be controlled through glutathionation. But when we looked for the same sorts of control in UCP1 through glutathionation, we found no evidence for that. So it kind of made sense that Ed Ciucciani and colleagues subsequently found a different means of control of those important thiol residues through redox modification. Now, there's been quite a lot of exciting recent research in a role for the adenine nucleotide translocator in mediating uncoupling. And the canonical role for ANT, of course, is the uptake of ADP into the mitochondrial matrix so that it can be phosphorylated to ATP by ATP synthase, and then the efflux of ATP so that it can be used to support energy-demanding reactions within the cytoplasm. But recent work through some really elegant approaches by Amber Berthollet and Yuri Kurashok's lab using patch clamping of the mitochondrial inner membrane has shown that ANT clearly causes a fatty acid-induced proton leak, and that the control of this leak through ANT is very similar to the control of the leak through UCP1, including fatty acid activation and the inhibition by purine nucleotides, including GDP. In this system, they found no evidence that UCP2 and UCP3 causes uncoupling, which is inconsistent with findings from our laboratory and others in other experimental systems. So it may be that the structural aspects or there are key components missing from this unique experimental system. And importantly, the last point there, they found that ADP-ATP exchange negatively regulates proton leak. So there's this reciprocal activity of the adenine nucleotide translocator for leak or ADP-ATP exchange. So it had actually previously been shown that ANT can play a role in proton leak uncoupling, and work in Martin Brand's laboratory showed in skeletal muscle that 1/2 to 2/3 of the basal proton leak is catalyzed by ANT, independent of ATP-ADP exchange. And work in my laboratory in UCP3 knockout mice found a compensatory type of increase in proton leak that we found was attributable to the activity of the adenine nucleotide translocator. Now, we've done quite a lot of research in mouse models to looking at the effects of UCP3, for example, in uncoupling in skeletal muscle, and some of our work from UCP3 transgenic mice, in which there's 2.5-fold higher levels of UCP3 protein in skeletal muscle, showed increased basal uncoupling, lower ROS emission, shown on the left-hand side, no change in the maximal respiratory characteristics of mitochondrial muscle, no change in mitochondrial content of that muscle, and in fact, increased complete oxidation of fatty acids in muscle and lower levels of fatty acyl carnitines in completely oxidized fatty acids from the mitochondria, which can be potentially damaging. Work from our laboratory and others showed that there is some protection from insulin resistance and obesity in mice. And the last point there, work from the Lombardi group in Naples showed that the progressive reduction of UCP3 levels in muscle results in insulin resistance and decreased fatty acid oxidation and signaling within the muscle. So at the human level in adults, it's been estimated that about 20% of resting metabolic rate or resting energy expenditure is due to skeletal muscle metabolism. This is roughly equivalent to the metabolic demands of the brain there, listed at 20% as well. But in the case of skeletal muscle, the reason for this large contribution is because skeletal muscle makes up over 40% of total body weight, whereas the brain, for example, 3%. So per gram tissue, of course, the brain is more metabolic, has higher energy demands than skeletal muscle. However, at the whole body level, energy balance, muscle makes a significant contribution. And of course, this goes up with physical activity and exercise. Now, how much of this is due to proton leak uncoupling? Well, it's been estimated that about half of resting muscle energy expenditure is due to proton leak uncoupling. And therefore, at the whole body level, proton leak uncoupling in skeletal muscle alone could account for 10% of resting metabolic rate. So another concept that should be covered, this slide seems to be cut off here, at least on my screen, but another concept that is important in the context of mitochondrial bioenergetics and in muscle and other tissues as well is the formation of skeletal muscle, is the formation of mitochondrial supercomplexes. It used to be thought that the multi-protein complex is one 2, 3, and 4 were distinct entities floating in the mitochondrial inner membrane. But recent research has shown that they form these higher order structures in the membrane with supercomplex formations of 1 and 3, 3 and 4, or when 1, 3, and 4 are all together. This is referred to as a respirosome. Importantly, complex 2, shown on the far left there, succinate dehydrogenase, does not contribute in the formation of supercomplexes. And for that reason, it's used often to appropriately normalize levels of supercomplexes within the electron transport chain. Now, the formation of supercomplexes is thought to be a very beneficial thing in the sense that this leads to increased oxidative capacity and perhaps also to efficiency and decreased reactive oxygen species emission from the electron transport chain, kind of a channeling of electrons within those supercomplexes. Now, our laboratory was the first to find that there's decreased mitochondrial supercomplexes in type 2 diabetes. And so we compared supercomplexes in skeletal muscle in individuals with obesity and diabetes versus those with obesity alone. And we found that there were significant reductions in complex 1, 3, and 4 containing supercomplexes, but no differences in the amount of complex 2 shown in the top right graph there. So there was clearly a decrease in overall supercomplex formation. And we also found there was a direct correlation of the formation of these supercomplexes with complex 1 and fatty acid supported respiration using high resolution respirometry in the permeabilized myofiber preparations. And on the right hand side there, showing an inverse correlation of muscle respiration with circulating levels of hemoglobin A1C in these patients. Now, subsequent to these findings, it was shown that exercise improves the formation of supercomplexes within skeletal muscle. And this diagram is from Francesca Amati's paper in Cell Metabolism in 2017. And it's also been shown that supercomplex formation decreases with aging. So now I'd like to move on to some of our translational research, looking at these concepts in the context of overall whole body bioenergetics and risk for obesity and capacity to lose weight quickly on a hypocaloric diet in a clinical weight loss program. So this is has been a collaboration with Ruth McPherson and Bob Dent for over 20 years now. And the main question that we have been exploring is, does variable oxphos efficiency, usually studied in muscle, play a role in diet sensitive versus diet resistant obesity? And for over 20 years, the same clinical intervention summarized in the diagram at the bottom there has been used in over 5,000 patients. So this has led to quite a platform for research in our collaborations here. So one of the findings that intrigued us very early on, and we've been pursuing in this context, is that weight loss in response to this 900 kilocalorie diet varies by 2 to 3-fold in adherent patients who are matched for age, sex, initial body weight, and in the absence of any other known factors that should affect weight loss, including certain medications, exercise, mobility, disease factors like thyroid hormone status, for example. And in the histograms in the top right there, you can see the variability in weight loss response at 6 weeks, 12 weeks, and 25 weeks. And what we typically do is compare those in the lowest versus the highest quintiles for rate of weight loss, the two red arrows in the top right there. Now, the questions of variability of weight loss response were addressed in a review that we had published in Metabolism last year, it's well known that there's variability in weight loss in response to hypocaloric diets, in response to exercise, in response to bariatric surgery and medications. But of course, the factors are complex. And what we have been mainly examining in our translational research are the impacts of genetics and metabolic efficiency mainly. But there is tremendous variation in metabolic rates was noted again by Herman Pomser and over 80 co-authors in a paper published in Science last year, in which they examined many data sets internationally using doubly labeled water data and rigorously controlling for lean body mass and other factors, and again pointing to variable energetic demands inter-individually. Now, in this slide, you can see the differences in the trajectories of weight loss in these two extremes, the uppermost quintile for weight loss versus the lowest quintile for weight loss. And we refer to those as the diet sensitive, or individuals who lose weight quickly on this 900 kilocalorie diet program, versus the diet resistant who lose weight slowly. And typically we define these groups at the 6-week time point when individuals are in this hypocaloric phase of the program, 900 kilocalories, and in which we are very closely monitoring their adherence to the program with attendance and blood biochemistry, return of empty packets, because this is a meal replacement program. And then after the individuals have completed the clinical weight loss program and they're in a weight stable state, we recruit them into our studies. And most of our studies involve muscle biopsies, in which we look at energetics. So with Bergstrom needle biopsies of about 150 to over 200 milligrams, usually from the vastus lateralis muscle, we can examine a wide range of characteristics and approaches, from respirometry to taking the satellite cells and differentiating those out into myotubes in culture, immunohistochemistry, Westerns, proteomics, gene expression, metabolomics, and lipidomics. And I'd like to show you some of the data in the next few slides. So our first observation with regard to mitochondrial proton leak uncoupling in the context of rapid versus slow weight loss was made back in 2002 when we published that paper in Diabetes. And in the top right, you can see the overall kinetics of proton leak. And we found that proton leak was 50% higher in the mitochondria isolated from the quadriceps muscle in the muscle mitochondria from the diet-sensitive individuals than the diet-resistant individuals. So the green line versus the red line, the oxygen used to support proton leak within those mitochondria. However, we wanted to take that further and look at proton leak and genes related to proton leak. And so in subsequent studies, we looked at gene expression profiling in skeletal muscle and also in whole blood before the individuals began the weight loss program. And we found increased expression of OXPHOS genes in the diet-sensitive individuals and the diet-resistant individuals, even in the whole blood samples taken before they entered the program. In other words, this was predictive of their subsequent outcome on the hypocaloric diet. And we found some distinct difference in skeletal muscle structure. We found that there was increased type 1 oxidative fibers and fiber hypertrophy in the diet-sensitive individuals. And those data are shown here. Higher amounts of type 1 fibers in the top left there in the diet-sensitive, shown as the black bar, versus the diet-resistant, the white bar, and in comparison to lean controls, shown as the hatched bar there. The fiber hypertrophy data are shown in the bottom left there, cross-sectional area of the fibers. So increased hypertrophy in the diet-sensitive individuals. And in the bottom right graph there, We found no differences in mitochondrial content between the two groups, but as others have shown, there's decreased mitochondrial content in these individuals with obesity compared to lean controls. And so then we wanted to determine whether this increased leak in diet sensitive could be detected in intact cells, intact muscle cells from the patient. So we took those satellite cells, differentiated them in culture, and found this was work of Brianne Thrush, a former postdoc in the lab. We found higher proton leak and glutathione redox in the diet sensitive than the diet resistant individuals. And then Brianne went on to conduct an in vivo and ex vivo study in which we fed two groups matched again for their initial body weight, age, sex. And most of our studies have been conducted like this one in women because females represent over 70% of our patients in the dietary program and also in the surgical program. And what we did here was provide the two groups with a high-fat meal, defined high-fat meal, biopsying before and after this high-fat meal. And we determined that there was higher capacity for fatty acid oxidation in skeletal muscle of the diet-sensitive individuals. And we also found some really intriguing data from unbiased plasma proteomics. This was Somologics proteomics. And some of the major hits are shown here. And on the top left there, you see the QQ plots, and three proteins deviated from that linear QQ plot, indicating statistical significance. And those three proteins are shown in the top row there with higher expression or higher levels of those proteins in the diet sensitive in each case, regardless of whether they were fasting. That's a T0 on the far left of each of those lines, or after, in the hours after the feeding of that high-fat meal. So those proteins included esterase D, which is also called S-formyl glutathione hydrolase, and the protein in the middle, there is heat shock protein 72, which is the chaperone protein often studied in muscle in the context of type 2 diabetes. We're very interested in that one. And the third one on the top right there is EIF5, a translation initiation factor. Then when we looked at the differential effects of the high-fat meal between the two groups, we saw significant increases in three proteins in the diet-resistant group, but no differences at fasting at baseline there. Now, the three proteins that there included AIP, or aryl hydrocarbon interacting protein, involved in signaling and protein targeting in cells. The one in the middle is PPID, which is peptidylprolyl isomerase D, involved in heat shock protein 72 binding, apoptosis, and lipid droplet biology. And the third was tyrosine protein kinase, FGR, involved in membrane signaling, inflammation, and immunity. And again, all of these were increased only in the diet-resistant group. Now, we were particularly interested in the heat shock protein 72 data, given its role in type 2 diabetes, the work of Mark Fabreo and Andrea Heavener and others. And so we determined whether there were significant correlations. We found that there were significant correlations in the circulating levels of heat shock protein 72 with muscle fiber respirometry, shown in the bottom row there, that there was a positive correlation between heat shock 72 levels in the blood and the respiratory characteristics of the muscle fibers, and also significant correlation with mitochondrial content in muscle, shown in the bottom right. So you can imagine that, with these observed differences in muscle fiber content, muscle diameter, muscle respiratory characteristics, that one of the questions that we've wanted to pursue for many years is do the two groups respond differently to an exercise intervention? So when Chantal Pileggi joined my lab as a postdoctoral fellow about 4.5 years ago, this is the project that she really led in our lab. And our hypothesis, or our hope really, was that exercise could be especially beneficial to those individuals with diet-resistant obesity, that this might be the missing link, or perhaps you could argue that these things are hardwired, and that a diet-resistant individual would be an exercise-resistant individual. And the findings are under consideration, under second review for publication, and hopefully we'll get some good news on this. This is major collaboration, a huge amount of work. Okay, so I'll just highlight some of the major findings from this large study. So we used this opportunity to dig deep into the data set from thousands of patients. Again, here we looked at women, and so here we looked at records from over 3,000 women, and we excluded those who were not deemed to be adherent. Then we divided the groups up into the quintiles for rate of weight loss at the 6-week time point, then removed any data sets where we had missing data, and then we looked at 114 matched pairs of women matched for age and initial body weight and BMI, and then looked at characteristics, blood biochemistry, and body composition characteristics using bioimpedance analysis. And we found that the diet-resistant individuals shown in the orange bars here had higher body fat, lower waist circumference, and lower fasting insulin values. But we wanted to use a more rigorous method for body composition. So we then went on to use DEXA scanning, and in 20 well-matched pairs of women. And we found that the diet-resistant individuals, again, had greater body fat and lower fat-free mass, and differences in the distribution of body fat, that the diet-resistant individuals had more lower body fat and less central adiposity than the diet-sensitive individuals. And given that the diet-sensitive individuals had higher waist circumference, fasting insulin, triglycerides, lower HDL, we are tentatively concluding that the diet-sensitive individuals are at greater risk for metabolic disease, something that we had never observed before. And so we're pursuing this in further research. Now, this slide just shows the exercise intervention. It was a supervised, progressive, combined aerobic and resistance exercise intervention, 6 weeks with biopsies and blood sampling before at baseline and post-exercise training, or PET, in 10 very well-matched women. And in the next few slides, I'll just quickly summarize the results obtained. So we found exercise preferentially improved body composition in the diet-resistant women, and showed, as shown in the top right there, lower resting metabolism and lower muscle strength, and again, lower content of type 1 fibers in the diet-resistant individuals. We found that exercise preferentially improved mitochondrial content in the diet-resistant individuals, and that exercise preferentially improved muscle fiber oxidative phosphorylation activity and permeabilized myofibers using high-resolution respirometry. And again, interestingly, just like we saw in isolated mitochondria and intact myotubes, we saw lower proton leak uncoupling in diet-resistant muscle. And here, permeabilized myofiber preparations at baseline, but not post-exercise training. Interestingly, in regard to proton leak uncoupling, we saw lower ANT protein levels in the muscle of the diet-resistant individuals, and as we've observed before, no differences in the absolute amount of UCP3 in skeletal muscle. Exercise increased mitochondrial supercomplex formation, but when we normalized to complex 2 levels, that's succinate dehydrogenase, as I've talked about earlier here, very important to do, those differences disappear. So we used the opportunity to collaborate with our colleagues who do some really great metabolomics, mass spec, metabolomic and lipidomic analyses. And we found interesting differences in glutathione precursors in the top left there, gamma-glutamylcysteine, and then post-exercise training differences in serine and histidine, and exercise effects on alpha-ketoglutarate, the bottom left there. Also very interesting to us in the metabolic perspective. I think I'll skip over this slide looking at ADP correlating with many of these metabolites, because I want to get to this last slide here in which I'm not sure if you can see it very well, but we found that exercise differentially altered serine sphingolipid muscle metabolite networks selectively in the diet-resistant versus diet-sensitive muscle. And the reason why this is significant is because serine is an important precursor for sphingomyelin, sphingolipids, including the ceramides, and ceramides are well known to cause insulin resistance within muscle. So the top two lines of this heat map are from the diet-sensitive individuals, and the bottom two lines from diet-resistant baseline and post-exercise training. So you can see the overall differences between the two groups, and you can see significant decreases in the red, the heat map, with exercise training selectively in the diet-resistant group compared to the diet-sensitive group. So in summary, this recent study has shown that there are distinct differences in body composition between diet-resistant and diet-sensitive individuals, differences in baseline energetics, and post-exercise training, there are selective improvements in metabolism, body composition in individuals with diet-resistant obesity. So I'll just wrap up now. The overall concluding statements that I could make include that the efficiency of energy transduction in cells is highly variable and responsive to the needs for ATP, responsive to the needs for thermogenesis in brown adipose tissue and for signaling. And this is largely due to proton leak uncoupling, and the mechanisms continue to be studied. There are complex subtypes of obesity, and the analysis of underlying mechanisms will help us devise more effective therapies. And a better understanding of proton leak uncoupling is relevant to many disease states beyond obesity and diabetes. So quickly, I'll just thank members of my laboratory, recent and current members, my collaborators in this recent research, and funding agencies. Thanks for your attention, and I hope to be able to answer any questions you might have. Thanks.
Thank you, Dr. Harper. This was a fascinating discussion. We have time for one quick question before we move on. I really appreciate how the impact of exercise is variable between the diet resistance and the diet sensitive. And we have a question that is asking you, what do you see the implications of next studies for you in terms of this diet resistant, diet sensitivity beyond the diabetes? What other diseases and approaches could you see this going?
Well, yeah, that's a great question. I think that a better understanding of the role of proton leak uncoupling in signaling within cells, redox signaling, a very complex area of cellular regulation will be important, and a better understanding of ANT-mediated proton leak is got to be very important too, because ANT represents about 10% of mitochondrial protein in many metabolic cell types, particularly in muscle. It represents 10% of mitochondrial protein. So
that's phenomenal. I can see the applications on antipsychotic dependent resistance and metabolic inductions, many areas of applications. We have several other questions, but unfortunately we're running a little bit out of the time. So I will connect to you directly and ask the questions and maybe send it to our members in the view. I thank you again very much for coming here today. It was a fascinating research, and please continue us keeping posted.
Thank you, Anna. I look forward to the rest of the conference. Thanks.
Thank you.